Planet. Today we will discuss about the enzymes. And enzymes, what we will discuss? In enzymes, we will discuss about their optimums. Optimums mean the value of either a temperature or pH where the activity of an enzyme is at its highest. Now, the maximum activity of an enzyme occur at a temperature is called as optimum temperature or optimum pH value. So this is what optimum temperature and optimum pH of value of an enzyme where the activity of an enzyme is at its best. For example, if we talk about the temperature value of human being, 37 degrees centigrade. It's the normal temperature of a human being where all the enzymes work at its best. Lower than this temperature, the activity will slow down, and higher than this temperature, the enzymes may be denatured. What is it? Denatured means the active site where the substrate binds may lose it. Like the shape of an enzyme is, if it is like this, if I say like this, so this shape is like, this is an active site, and this active site may be denatured. Now the shape may went like this. If the shape is like this, so the substrate, like substrate, may be of this shape and it may not bind here. Once it will not bind here, what will happen? It will not be able to perform its function. So at higher temperature, it may denature. So if we see the temperature graph, so at the start, if we see, the rate of reaction will increase, and at certain temperature it will be maximum. For example, this is a temperature and it is an optimum temperature. After that, the enzyme activity will reduce and may become the zero. Clear? So this temperature is called as an optimum temperature, and after that it will retard. So here, why optimum temperature is required? For the highest activity or the best performance of an enzyme, we need this one. What we need? What do we need? We need the optimum temperature. Same is the case with the pH value. So optimum pH value, each enzyme perform better at certain pH value. For example, in like gastric area, like the stomach, in stomach, the pH value is about 2.0, where in acidic medium, germs are killed and the performance of the enzymes are the best, like the pepsin. So it will be at its best. Okay, so if it is less or more acidic, it may disturb the stomach area. So this is maximum pH value, and the maximum pH value or the pH where the enzyme activity is at its highest, is at its best. So this is what we call it as the optimum temperature, or optimum pH here and optimum temperature. So same type of graph we can observe, but here it will be the maximum value for the pH, but the temperature graph is here better than the other graph. So let's discuss these pH values of different enzymes they are performing at its best. As for the exam definition, the optimum is the temperature or pH at which an enzyme has its highest activity. Now, what will happen if the optimum is not reached? For most human enzymes, 37 degrees is the optimum temperature. Below the optimum temperature, molecules move more slowly, fewer successful collisions occur, like they will collide with each other. As a result, something will happen. Enzyme activity decreases if it's below the optimum temperature. If it is above the optimum temperature, enzymes begins to denature. The active site changes shape. The substrate can no longer fit properly. Activity rapidly decreases. As I have shown you, the activity increases gradually up to the optimum if you see the optimum graph, temperature graph. After the optimum, activity drops sharply because of denaturation. Okay. Now look at the optimum pH. Pepsin, location is stomach. Optimum pH is about 2. Amylase, it's in the mouth, small intestine, and about 7 is the optimum pH. Trypsin, small intestine, and about 8 is the optimum pH. Now, why it's necessary? The shape of enzyme's active site depends on pH. If pH becomes too acidic or too alkaline, the enzyme may denature or its active site may change shape. The substrate can no longer bind effectively. So now let's discuss some of the terms for IGCSE. Active site, the region of an enzyme where the substrate binds. It's an active site. And the substrate, the molecule upon which the enzyme acts. And enzyme-substrate complex, the temperature or temporary complex formed when a substrate binds to an enzyme. And denaturation, a permanent change in the shape of an enzyme's active site caused by high temperature or extreme pH. So the exam definition for the denaturation is the permanent alteration of an enzyme's structure so that it can no longer function.